Welcome back to Chapter 6. Now we're going to do Part 3, really focusing on a molecule called ATP. ATP stands for adenosine triphosphate, and we're going to learn way more about ATP than you ever thought you'd want to learn. But ATP is extremely important for a life, and we're going to find out just why it's so important as we go through this part of the lecture. So, ATP, again, adenosine triphosphate, takes and powers cellular work by coupling exergonic reactions and endergonic reactions. And this is in order for cells to do work. And there's three main kinds of work that a cell does. It does chemical work, it does transport work, and it does mechanical work. And we're going to go through all of those eventually. But we're going to start out here by really getting familiar with the concept of ATP and learning a lot of the structures here. So, ATP does its work by doing something called energy coupling. Again, it uses exergonic reactions to drive endergonic reactions. And almost every energy coupling that happens in cells is mediated by ATP. If it's not by ATP, it's something like ATP. So you can kind of think about ATP, and again, that's adenosine triphosphate, as a charged battery. And as you have this charged battery, and if it releases its energy, you have a dead battery. But if it's a rechargeable battery, which ATP is, you can recharge it and make it again. Let's take a slightly closer look at the details here. Again, we're going to get into a lot of details, but for now, here's our cartoon of ATP, and it's got 1, 2, 3 phosphates there on the end. Water comes in, and that's right. If water is coming in to break something apart, that's hydrolysis. So it breaks off one of those phosphates, and now we have ADP, and ADP, it is adenosine diphosphate. That's our dead battery. If we want to recharge that battery, we have to put this phosphate back on the end, and that does release energy. We're building something, so it does require energy to do this, and we'll talk a little bit about how that happens later. But when it does happen, you've got your charged battery again. Let's take a look at the structure now. Adenosine triphosphate is composed of ribose sugar, right there. It's a 5-carbon sugar. 1, 2, 3, 4, and you can see that 5th one right there. There's carbons there at those vertices. And it's made up of adenine. This is a nitrogenous base. We learned about this in chemistry. So a nitrogenous base, like the kinds you find in DNA, this is adenine. This is exactly the nitrogenous base that you would find in a DNA or RNA molecule. So what's different about this is that it has 1, 2, 3 phosphate groups. And those phosphate groups hanging off the end there, that just has a lot of potential energy, like high potential energy. So in addition to the ATP, it is used to make RNA, and we're being really specific here about RNA because as long as you've got adenine, like adenine can be found in DNA and RNA. But ribose, that's only found in RNA. So ribonucleic acid, it's named after that sugar ribose there. So this can also be used to build RNA. And we're focusing now on the structure of the phosphate groups over here. And ATP in the cell, most of the hydroxyl groups that you have are going to be in the ionized form. So O negative, like that's got the ion. The hydrogen is stepped away. It's not here right now, so it's in its ionic form. Again, the bonds between the phosphate, those are the key things here, and those can be broken by hydrolysis, as I said before. ATP hydrolysis releases energy, and that energy is going to be used to drive our other reactions. And now it's ADP, adenosine diphosphate. And we have something also hanging off here called inorganic phosphate, and that's always symbolized as P sub I. Often it's symbolized in a circle. So you see that P is in a little circle right there with a little lowercase I below it. That means it's inorganic. That means that it's not attached to a carbon. Remember that carbon, that's what kind of qualifies things as being organic or not. So if it's all by itself, there's no carbon, it's inorganic. The energy that's released, the energy released comes from the change in the chemical state from having high free energy to low free energy, not from the phosphate bonds themselves. It's this idea of the whole molecules kind of change its energy state. ATP hydrolysis releases a lot of energy, and it's due to the repulsive force, like repelling, right, of the 3 negatively charged phosphate groups. Those negative charges are kind of in tension, and when the triphosphate tail of the ATP becomes, when it has all 3 of them on there, it's like a spring, and that spring just wants to push off. So it releases a lot of energy when that 3rd phosphate is let go. All chemical work, almost all, I should say, chemical work is powered by this ATP hydrolysis. And energy released by this exergonic reaction helps drive those endergonic reactions. ATP drives these endergonic reactions by phosphorylation. So remember, we had our phosphate group that was on ATP. But what's going to happen, it's going to get transferred. That phosphate group that was released is going to get transferred to another molecule like a reactant. And when that reactant has the phosphate group on it, we say that that recipient molecule is called the phosphorylated intermediate. That kind of gives you the idea that that intermediate stage is not going to last very long. And it's not going to stay phosphorylated. It just stays that way for a little while. Overall, the coupled reactions are exergonic, meaning overall energy is being released. We are following the laws of thermodynamics. Let's take a look at what happens in various types of work. And specifically, we're going to look at energy coupling driving chemical work. And this is just an overview. I'm going to show you the step-by-step over the course of the next 3 slides. But kind of just wanted you to orient to what you're seeing here as we step you through. We've got our reactants and our products, and we've got ATP coming in, and we're going to look a little bit at numbers. I'm not going to make you calculate numbers per se. It's just a subtraction. It's not that hard to calculate numbers if you were given numbers, but you don't have to look it up quite like you would maybe in a physics class or in a chemistry class. So let's take a look at the first step. This is where we've got glutamic acid, that's the pink triangle, and we're going to convert glutamic acid and ammonia, combine those and create something called glutamine. And by itself, this is an endergonic reaction. Remember, endergonic reactions have a positive delta G. That means that it requires an energy input of 3.4 kcals per mole, and so that's not a spontaneous reaction. So we need some help. And the way that we're going to get this help is by using ATP. So instead of just joining the glutamic acid with the ammonia, what we're going to do is an intermediate step. Glutamic acid is going to combine with ATP to form this phosphorylated intermediate. And you can actually see that phosphate group is now connected onto the glutamic acid. In step 2, and we do have this release of ADP. Sorry, I meant to mention that. In step 2, we're going to add in our ammonia, and the ammonia is going to come on, and it's going to force the release of that phosphate group off of the glutamic acid. It's going to force that release, and we're going to join those 2 together, and now we have glutamine plus some ADP plus some inorganic phosphate. So if we were going to do this calculation, we would do it like this. Glutamic acid and ammonia, remember they had a delta G of plus or positive 3.4 kcals per mole. Releasing phosphate from ATP, the delta G of ATP, is negative 7.3 kcals per mole. And so we have this net delta G of minus or negative 3.9 kcals per mole, right? So if you add those up together, you get this negative number. And that means that this can happen spontaneously. As long as there's ATP around, you can make glutamate from glutamic acid and ammonia spontaneously. So how does ATP work? I've been foreshadowing this a little bit using terms already. And we're going to be talking about ATP hydrolysis, right? That's the thing. We're kind of lopping off that third phosphate. If we're trying to get ATP to do transport work or mechanical work, as opposed to the chemical work that we just looked at, we're still going to need ATP hydrolysis. But instead of latching onto a product and to make a reactant, we're actually going to use that inorganic phosphate that's lopped off of the ATP to change the shape of a protein. So, and that can also change its ability to bind to other molecules. So we'll still talk about phosphorylated intermediates, but it could be a non-covalent bond between ATP and a protein. It depends. Let's take a look at a couple of examples here. So, the top one that we're looking at is a transport protein. Now, remember, transport proteins are found in plasma membranes, and we learned that in the last chapter. And so what's going to happen here is this transport protein is going to grab onto the thing that is going to transport across the membrane, but it can't change its shape unless it's phosphorylated. So ATP is going to loan one of its little phosphate bits, and it's going to phosphorylate this to make this phosphorylated intermediate, which is going to cause it to change its shape and release the solute into the other side of the plasma membrane. Notice that it's going against its concentration gradient. So maybe you might remember what that's called when molecules move against their concentration gradient from an area of low concentration to an area of high concentration. If you don't remember that, you might want to go back and look that up. We can also use ATP to help with mechanical work. So ATP is going to bind non-covalently to these motor proteins. We talked a little bit about motor proteins in a previous chapter. Motor proteins are basically proteins that move or walk along a cytoskeleton. That's one major thing that it does. Also, there's lots of motor proteins in your muscles. So this mechanical work of moving this molecule along a cytoskeleton track or a highway inside your cells requires ATP. It's going to bind non-covalently to that motor protein. And then when ATP is then hydrolyzed, it causes the change of shape at that point. So ATP as a whole binds, not just the inorganic phosphate. Then the ATP loses its phosphate and becomes ADP, and that little leg moves forward. It's really cool. That's transport work, moving things across a membrane, and mechanical work, the act of walking of a motor protein along a cytoskeleton track. Now, ATP is regenerated as well, and so it's renewable like that battery that I was talking to you about before. All we have to do is add a phosphate group. So you're going to add that phosphate group to ADP, and that energy is going to come from catabolic reactions. Remember what catabolic reactions are? If you're having trouble remembering, you might want to rewind or look at the previous video in this chapter and remind yourself of what a catabolic reaction is. ATP goes through this cycle over and over again. It's a revolving door of forming ATP from ADP and then forming more ATP and then releasing that energy and so forth and so on. So very renewable. All you need is a little bit of energy, 7.3 kcals, right? That was the amount of energy that is released. It's also the energy that is needed to add that phosphate group back onto the ADP to make ATP. Well, that's the end of part 3. We'll continue this lecture in part 4 in the next video.